Good afternoon, everyone. So, we will start with today's tutorial session for week 8 lecture videos. Start presenting. I am audible to all, right? Okay, okay. Um, So, last week we looked at few chromatographic techniques like hydrophobic interaction chromatography, and ion exchange chromatography. And this week, we will be looking at gel filtration or size exclusion chromatography and affinity chromatography. Okay. So in size exclusion chromatography, what happens is size exclusion chromatography is also known as gel filtration and or gel permeation chromatography. So uh, abbreviated form is mostly used in regular uses like either SEC or gel filtration. Okay, so it separates molecules based on the differences in size as the whole mixture of molecules passes through a resin, SEC resin packed in a column. We will look at how, like, what can be the different types of resins, how we can pack it and all. Okay. Unlike techniques such as IEX is ion exchange chromatography, or AC means affinity chromatography, molecules in size exclusion chromatography do not bind to the resin or matrix. Okay. So what happens here is the buffer composition does not directly affect the resolution. Resolution last week we saw like how well you can separate two different molecules. How, like resolution of a matrix depends on how well the matrix can separate the mixture of molecules, or how prominent two peaks are, or like what is how well separated two peaks are from each other. Okay. In a chromatograph. And so what happens here is size exclusion chromatography. You just have to put in your buffer and the mixture of molecules, and basically what the protein of your interest or the molecule or complex, suppose, or the like entity of your interest should be stable in that buffer condition, and you put it there, and based on the particular size of your sample of interest or entity of your interest, the thing will elute out at a certain volume based on its size. Okay. Hmm. And the significant advantage of doing SEC is the conditions can be varied to suit the type of sample or the requirements for further purification, analysis, or storage without altering the separation. Okay, because here what is the basis itself is separation on the basis of charge. So you can use whatever buffer you want to, whatever pH you want to. Okay, because here ideally in SEC there should not be like uh, there is no interaction between the matrix and the mobile phase. Okay, basically we have to choose the mobile phase based on how stable or whatever pH or salt condition your protein of interest is stable in. Okay, and then you just pass through the column, and it will be separated based on the charge, but again sorry, based on the size. But yes, one thing we should be noting here is the size exclusion chromatography columns that comes. Okay, so the pores can be different, so we have to take care of the pressure limits of the column and everything, so those can be taken care of. But you can use any type of buffer condition, because ideally it should not react with the matrix. Okay, or there is no interaction with the matrix, or the sample of interest or the entity of your interest that you want to purify, that also doesn't interact with the, or react has any sort of interaction with the matrix. It will just pass through the different kinds of pores present in the resin. Okay. Now, SEC is a highly versatile separation technique. It is well suited for biomolecules sensitive to changes in pH, concentration of metal ions or cofactors, or harsh environmental conditions. Okay. It can be separated in the presence of essential ions, cofactors, detergents, urea or guanidine hydrochloride at high or low ionic strength. Urea or guanidine hydrochloride are mostly, detergent is also one of them, like how we saw in SDS page electrophoresis, right? That we use SDS, this detergent, to denature the protein samples that we want to run on the gel. So urea and guanidine hydrochloride are also denaturing reagents, okay, which is used to denature the protein and shun like in like stop or reduce the interactions taking place between different kinds of units, okay, of subunits of a protein. This SEC can be performed within a wide temperature range covering the requirements of most experiments, and the most commonly used temperature range is 4 degrees to 30 degrees Celsius. Okay. But there are SEC columns which enable separations at even higher temperatures. That again depends on what is the matrix or the what is the resin composition. What is the composition of the resin of the SEC being used at higher temperatures, and the pressure limits, etc., would also be variable. Okay. And purified biomolecules may be collected in any chosen buffer. Okay, that is what I was talking about. Basically, you have to take care of the buffer in which your sample of interest or the complex or entity of your interest that you want to purify is stable. Okay. Now, SEC resins are usually porous matrix of chemically and physically stable spherical properties. Okay, with properties that minimize the adsorption of biomolecules. There should be no interaction with the sample or the mobile phase passing through it. Okay, so what happens is it is a packed bed equilibrated with an appropriate buffer, and this buffer is ideally the same one through like in which your sample is present. But the very first thing, how we looked at the previous chromatography techniques, you have to first wash the resin or column, whatever chromatographic column you have to use. Okay, in case of SEC, you first wash it using like filtered water, okay, milky water or like double distilled water and degassed water. And then what happens is you equilibrate it with buffer, the buffer in which your sample is present, and then you pass your sample which is present in the same buffer, equilibration buffer. Okay. Now packed bed equilibrated with buffer, what happens when we equilibrate the resin with the buffer? It fills the matrix pores and the space between the particles. Okay. Now, this liquid inside the pores or the equilibration buffer inside the pores or the stationary phase, the resin or matrix is the stationary phase, is in equilibrium with the liquid or buffer, the equilibration buffer or the mobile phase outside the particles. Okay, when we are equilibrating it with an appropriate volume. Okay. And sample components are eluted isocritically. This is important because in the ion exchange chromatography and hydrophobic interaction chromatography also, we were looking at gradient elution, step elution, right? Like wherein we are changing the composition of the buffer so that the interaction of the molecules or biomolecules changes with the matrix. Okay, matrix components. Here there is no need to, because there is no interaction, or there ideally should be no interaction with the matrix. Okay, just we are separating it via the like through the different sized pores and the based on the size, like properties, like different sizes of the biomolecular mix. Okay, so isocritically means it's the same throughout, the composition of the buffer is same throughout. Okay, so the buffer composition remains constant throughout the separation. So there is no need to use different buffers during the separation. Only one buffer you just pass through through the column, that will be fine, like enough. And a wash step is sometimes used, not sometimes, it is used using the running buffer often included at the end of a separation. Now, first you equilibrated it, okay, and then you passed your sample of interest through the resin. Next, you wash it using the same buffer, because what might happen is, suppose there might be some part, there might be, see, in an ideal case, suppose there might, in the porous matrix, what happens is smaller sized particles get inside the pores, and the larger particles which cannot get into the pores, they move, like, elute out faster, okay, as compared to the smaller particles. Now, this wash step is included because some smaller particles which might just remain inside the pores only, which might not have come out properly. So this last wash step using the same buffer is used at the end of a separation, so that everything comes out. Now, why is this necessary? Because, see, SEC separation is based on how like how good the resin is, and how free the pores of the resins are, right? Because that is what will eventually help in separating the uh, separating your protein of interest, or purifying the protein of your interest, or any other sample of your interest, right? So if those are clogged, okay, so those need to be free of any possible particles after after separating your protein of interest or your sample of interest, okay. So this wash step will remove molecules that might have been retained on the column and prepares the column for a new run, okay. And this is size exclusion chromatography can be performed directly after ion exchange chromatography, hydrophobic interaction chromatography, or affinity chromatography, because the buffer composition will not affect the final separation. Okay. Now see, this is the matrix we were talking about, and here this is an electron micrograph of the same thing. See, there are pores of varying sizes. Okay, so the larger molecules won't be able to get inside these pores. Now, suppose this is a mixture. Okay, here, this one having this yellow particle, this orange colored particles, these largest red colored particles. Okay, in your mixture. Now, what will happen is, in this porous matrix, when these pass through this porous matrix, the smaller ones will definitely get inside and get trapped, or like their passing or their movement will slow down, because they will be moving through the pores. And these, and some of the, like, most of the smaller ones will get inside the pores. These medium orange ones, some of it will get inside, some of it will try to pass through the columns, but as in through the pores. But these larger ones cannot enter the porous matrix, and they will be passing through and moving faster than the other two types of molecules here. Okay. So in this example, there are three ones: yellow, orange, and red. Now see, this is the separation that is being shown here on the matrix, on the resin. So this is, you have applied the sample here, or you have loaded your sample. Okay. And now the yellow ones, the orange ones, the red ones, right? The yellow ones being the smallest has entered the pores. So it is going through each pore and moving through, like, getting inside the pores and moving through the matrix. So it will take the longest time. And then, as compared to that, orange one is slightly faster, but the fastest one is the largest particle, okay, which is the red one here. And then over the time, slowly, so this is like slowly the things are passing through the matrix, and over the time, see, this will reach the end, like the largest red one will elute at the like uh, will reach the end faster, or elute out faster, as compared to the orange one, which will again elute out faster as compared to the yellow one, the smallest one. Now, one thing here that needs to, you see like the thickness of the thing. Okay. Now in the last class, we read about adsorption, distribution coefficient, okay, or adsorption coefficient. So the more the interaction with the matrix, the longer time the molecule will take to elute out of the column, okay, or elute out of the matrix, because it is interacting with the matrix, getting absorbed properly, okay. Now what also happens is, suppose this, see the band broadening in this case, for the smaller solute here, smaller particle here is broadest as compared to this will just, it won't react, or it won't stay in the matrix or column. Here there is no adsorption with the matrix, it is just like through the pores, this is passing through the pores. So there is a zone or band broadening, okay, of the smallest particles, because it is going through the things slowly over the time, okay, along with the buffer. But this one, the red one, hasn't entered the pores at all, and it is just passing through. So everything will it won't really take time, or like take time to pass through the matrix pores, and just elute out the fastest with a very thinner uh, uh, zone broadening, okay, or in general like thinner band as compared to the smallest one. See, the band is higher because it is taking a longer time to go through the pores of the matrix, okay. And this is the elution profile or the chromatogram. This is the absorbance, this is your column volumes, which means the buffer at which your things are, or your molecules in the mixture are coming out. So you inject your sample here, okay, and now this is your high molecular weight, the red one, which comes out first, followed by this intermediate molecular weight, the orange ones, that comes out next, and then like in the end, this yellow one, or the smallest particles, come out, the low molecular weight particles come out. Now we will eventually look what this VO, VE and VT mean. Okay, VO is the void volume, VE is the elution volume, and VT is the total volume, okay, of the column in question, okay. And definitely you see here, you first equilibrate your sample, and then sorry, equilibrate your matrix or the resin, and then you apply your sample or inject your sample, okay. Now, size exclusion chromatography can be preparative, analytical. Okay, so in preparative size exclusion chromatography, what happens? High resolution size based separation of biomolecules with fractionation. Okay, now when we are talking about fractionation, so so fractionation means the same thing like for the previous chromatographic techniques we looked at. Suppose this is your column, and then this will be eluting out, right? Now you will be collecting the fractions. So this is what we call as fractionation. Okay, like one by one. Suppose now this wall. Now what happens is, uh, based on how good the separation is, or what is the gradient applied, not in SSC or in general, I'm talking about where gradient separation or step elution or gradient elution takes place. And here in SSC also, what happens is you you have to collect what is being eluted out, right? So so that is collected in, suppose maybe you start collecting 200 microliters or 500 microliters or 1 ml of 1 ml volume of fractions. Okay, fractions means the same sample that is being eluted out of the column. Okay, or people take 2 ml, people can also collect 1.5 to 2 ml of fractions. Okay, depending on like what is your protein of interest, how good is the resolution of the matrix. So depending on the properties of the resin, and where exactly your molecule of interest is coming out or eluting out of the column, depending on that the fractionation volume can change. Okay, and what is the buffer conditions, and what is the program that you have set given the whole system, so that the whole process come, like how good is the flow rate, or how much is the flow rate. So based on these parameters you set your elution volume and get the fractions. So this whole elution and fractionation is basically collecting particular fractions, or eluted out fractions of a particular volume. Okay. Yeah, isolates one or more components of a sample, preparative size exclusion chromatography. The separated components can directly be transferred to a suitable buffer for assay or storage. Okay, so usually SEC is the like final step of purification, and which can be used directly for your downstream assays or in any kind of experiments you want to perform, or you can directly store the molecule of interest that you have separated and then use it later for your experiments. And in preparative SEC, what happens is you apply small sample volumes, which uh, which is around 0.5% to 4% of the total column volume of the resin, SEC resin, applied at low flow rates using long columns, often 60 centimeters or longer. Okay. And particle size commonly is greater than or larger than 12 micrometers, providing low column back pressure. And the resolution of preparative SEC varies from high to more moderate depending on the particle size. And for optimum results, samples with few components should be used, like those partly purified by other chromatographic techniques. That's what the previous slides also we saw, that after ion exchange or after affinity chromatography or after hydrophobic interaction chromatography, you basically uh, like f- do SEC, so that that should be the last step ideally. Okay, wherein you get the like purest form of your protein or your any sample of interest or complex being talked about. Okay, so ideally if there are two, suppose if initially for other ion exchange chromatography, hydrophobic interaction chromatography, we were seeing like a mixture of like the whole cells were being lysed, things were centrifuged, the lysate was loaded, the lysate had too many particles, right, too many molecules which are not of our interest. Okay, so if all those things are applied directly in SEC, then because this is based on the separation of the size, okay, like based on like based on the sizes, and which can enter into the porous matrix, pores of the matrix, then what will happen is like there might be like a lot of molecules might hamper the final result. Okay, a lot of molecules of different sizes can hamper the result or the resolution of the SEC. Okay, so it's for optimum results or for getting a well resolved SEC peak, uh, what we should do is we should have samples with few components or partly which have been previously purified using other chromatographic techniques, and finally you use SEC for the final step of purification. It is well suited for the final polishing step in the purification scheme. Okay. Now see, this is the same thing that is being shown here. You inject your sample after equilibration definitely, and this is your high molecular weight proteins will elute out first, followed by intermediate molecular weight proteins, and the low molecular weight proteins which have entered into the pores of the matrix, that will elute out at the end. Okay. Now another is analytical SAC. What happens here is this is high resolution size based separation without fractionation usually. So what happens is you are analyzing, okay, suppose whether it's a polymer, oligomer, I mean, if, whether it is whether your suppose protein of interest is oligomerizing, etc. You don't need to check, you don't need the sample there, you're just analyzing your suppose you have formed a complex or not. If something has formed a complex or not, then you want to analyze the thing. Okay, how well the things are, or where exactly the things are coming out, or whether it's forming aggregates or not, like what is the property or characteristic, how stable is your protein or complex. Okay, after a certain, suppose after whether it's stable in room temperature or in 4 degrees, like you want to see the stability, check the stability of the complex you have formed, etc. So you can just run through your SEC column and analyze the thing. Now analysis often is connected to high selective detectors like mass spectrometers, photodiode arrays, like multi-angle light scattering or fluorescence detectors. Okay, this is performed to check sample quality or to study biomolecular properties. Okay, here, small sample volumes are used, like really less sample volumes are applied. 0.3% to 0.5% of the CV. CV is the total column volume of the matrix, which is applied at low flow rates using long columns, typically 30 centimeters. The particle size is commonly 4 to 12 micrometers, providing high resolution. This can also be used for rapid purity checks and screening. Shorter columns of typically 15 centimeters provide adequate resolution, and short cycle times together with small volume and low buffer consumption. Okay, so if we are just for quality check and all, these analytical SEC is performed. Okay, and for properly purifying your sample of interest or protein of interest, you use the preparative SEC, where you have to collect the sample that is coming out of the resin and use it for further downstream experimental assays or like for storage only. Okay, you can store it at we use like for proteins. There are proteins when we purify proteins, usually what we do is we immediately after SEC, that is the last step. Before that, we can use different chromatographic techniques, and then after SEC, we just flash freeze it using liquid nitrogen, so that we just arrest the thing there, and then we store it at minus 80 degrees Celsius in aliquots, so that we don't have to freeze for the sample multiple times in small aliquots. And then we just take out one aliquot or how much ever is required. That many number of aliquots are taken out, and we perform the experiments accordingly. Okay. Yeah, one thing to keep in mind is shorter columns typically used for purity check or screening give lower resolution than longer columns. Okay, because see the separation, the while the things are interacting, uh, here it's not interacting, things are passing through the porous matrix, but for just for shorter columns, that resolution will definitely be lesser as compared to longer columns, which are giving more time for the constituent molecules of your sample, or of your of the mixture that you have applied of the sample in the sample. That sample, the biomolecules take are getting enough time as well as enough uh, distance to really interact, like or pass through the pores, or whether to pass without getting into the pores of the matrix. Okay, so longer columns will have higher resolution. Okay, the columns which are used for purity check or screening. Another use for SEC is desalting and buffer exchange. So what happens is, suppose suppose you have your protein of interest, or a nucleic acid of your interest, along with you have set a reaction. Now you want only the protein of your interest from there, or suppose you want your nucleic acid, or the some DNA or RNA of your interest from that whole mixture that you have used in your experiment or the reaction. Now, for suppose some downstream experiment, you don't need that buffer composition. Now, how will you really remove the salts or different kinds of salts from there? So, what you do is you pass the whole mixture through an SEC column, SEC resin, and then salts being extremely small in size as compared to your complex or your nucleic acid or your protein of interest, what will happen is your protein of interest will elute out first, definitely, because those won't be able to really enter into all the pores of the matrix, as compared to the salts, which are really small as compared to the main biomolecule of your interest, and those will come out at the end. Okay, so group separation, where small molecules like salts or free labels separated from a group of larger molecules. Okay, and samples can be prepared for storage or for other chromatographic techniques and assays. Okay, it depends, whatever is the downstream experiment or application you want to have. Based on that, we need to sometimes, suppose what happens is, recently, suppose in some experiments, what we have to do is, we have to go for mass spectrometry for determining the mass of a solute, okay, or mass of a sample. Now, for mass spectrometry of a nucleic acid, all these buffers that it is in the solution, that the tRNA is in, or the nucleic acid is in, what happens is, or the RNA basically is in, that has to be removed. Otherwise, it will hamper or affect the mass spectrometry results. So for that, we can pass it through desalting columns, where the salts come out, either salts come out, or salts get separated from the molecule of our interest, and then we can get only the molecule of our interest and send it for mass spectrometry. Okay, so like this is just an example, so there can be other examples as well. Now for desalting and buffer exchange, what happens is larger sample volumes, or that is desalting. In buffer exchange, what happens is kind of the same thing. Suppose for the next experiment you need certain other conditions, certain some other buffers, or suppose even in case of storage you don't need the presence of a high amount of salt or something, then you basically exchange the buffer or change the salt conditions, salt concentration in the buffer, uh, using the like either these resins or some other concentrator or filters, wherein you basically uh, exchange the buffers that with the like exchange the buffer that the solution or the sample is already in, with the buffer that will be used in the downstream or experiments or for your uh, storage or other applications. Okay. So in case of desalting and buffer exchange, larger sample volumes are used, up to 30% of the total column volume, that can be applied at high flow rates using broad, short columns. Particle size is relatively large, providing low column back pressure and high flow rates. Okay. Because if the particle size is large, it won't be entering the pores of the matrix, right? So it will pass through without getting inside the pores. So you can keep the flow rates higher. If the things are smaller and things are getting inside the pores, then what might happen is if high flow rates can damage the pores of the matrix. Okay, because the smaller solutes or smaller molecules can get inside the pores, and high flow rates can uh, damage the size of the pores or the integrate, like the basically the pore sizes. It can damage that pore size, thereby affecting the resolution and further usage of that particular resin or column. So here, see what happens is this is your protein of interest that will elute out first, um, and then the salts will elute out later. So large molecules have no or limited access to the resin pores and are thus eluted in or just after the void volume. So just to give it, void volume is what happens is your pores are packed. Okay, I mean the matrix is packed, resin is packed. Void volume is the one which is outside the matrix volume. Okay, whatever spaces are there outside the pores, that is the void volume through which your larger molecules can pass through, because it cannot enter the pores. You saw the pore like electron micrograph of the pores, right? So it is like designed in a way that larger molecules won't be able to enter inside. So usually what happens in the void volume only, these larger sample for interest or proteins elute out. And what happens is one column volume is basically the whole whatever volume of the packed resin is there. Now the salts, when one column volume is the salts elute out almost near the one column when this one column volume of buffer has eluted out. Okay, because salts are being smaller, these salts can get inside the pores and take much longer time to come out. And as soon as like suppose a column is 30 centimeters long or 60 centimeters long, so 60 ml, as soon as 60 ml or nearly 55 to 60 ml starts to come out, then you see the peak for salts. That's when the salt also starts to come out. Okay, because along with the mobile phase, it is also coming out slowly, coming down slowly, but it is moving like getting inside all the pores of the resin and then coming out. Okay, so it elutes out almost near one column volume of the matrix. Column volume means if the column volume like matrix is 30 centimeters, suppose, so 1 CV will be 30 ml. Okay. Small molecules such as salts have full access to the pores and move down the column more slowly. So these molecules usually elute just before 1 CV of buffer has passed through the column. Okay, here, that is what is being shown here. And see, UV proteins, we measure the concentration or absorbance of proteins at 280 nanometers. Why? Because of the presence of aromatic amino acids that are present in proteins, okay, which given absorbance, specifically tryptophan shows the highest absorbance at 280 nanometers. There is another thing that we get to see in the chromatogram, that is conductivity. So when suppose when you equilibrate your column, what will happen is it will give you a kind of sigmoidal thing wherein slowly your conductivity will increase, but when the column is equilibrated with the same buffer, it will reach a plateau. Okay, that means your column is equilibrated. So similarly, based on what is the ionic strength of the buffer being eluted out of, or the fraction that is being eluted out, depending on that, if the salts are coming out, then slowly see the conductivity will increase. This is the conductivity graph, and then it will as all the salts are coming out, it will again go down. Okay, this can also be measured here or can be seen in the chromatogram. Okay, now see here, molecules that are larger than the largest pores in the matrix cannot enter the matrix. So these molecules are eluted together in the void volume or V naught as they pass directly through the column. This one. Okay. This is the void volume wherein it cannot enter the pores and it will just pass through the column and elute out first. So this will come out in the void volume. For a well-packed column, the void volume is equivalent to approximately 30% of the total column volume. Okay. And molecules with partial access to the like intermediate molecular weights sample, what happens in those cases, like molecule, they have partial access to the pores of the matrix, and these are separated and elute from the column in order of decreasing size, like this one comes out, then this one comes out. Okay. Then small molecules such as salts have full access to the pores and are not separated when they move down the column. Okay. These molecules usually elute at the total liquid volume. Okay, this is VT. Hmm? Slightly before 1 CV, or which is the column volume of buffer has passed to the column. This is what we looked at in the previous graph also. Salts are eluting out just before 1 CV. Okay. So V naught is the void volume. VT is the total liquid volume that is passing through. And this is the geometrical column volume. This is the actual available volume for the liquid or mobile phase to pass through. And pore volume is VI. Okay, whatever pore is present, that is VI. Okay. See, this is your resin or matrix that is having pores. Okay. Now void volume is this one, the blue region. Okay. Like neglecting the pores, whatever volume is present in the whole column, that is your V naught, void volume. And when we talk about column volume or the total bed volume, that includes the pores as well as these spaces between the pores or outside the pores. Okay. So this total thing the, is the total bed volume or VC. So what will be the pore volume then? Pore volume will be, if we want to calculate only the pore volume, which is VI in this case, this is VC minus volume of the matrix minus the void volume. Okay. This will be the whole pore volume, or in general, it can directly be simply the total bed volume minus the void volume. It will give you the volume of the pores. Okay. Actually, V matrix here, what happens here is, like if we really get into the nuances, what happens is matrix is also made, like the pores are made up of the matrix, right? So it has some amount of thickness or some amount of occupancy, right, in the whole resin. So if we remove that particular matrix thickness or matrix volume that is being occupied in the whole resin, then if we remove that also, then the available pore or the available volume of the space that is there inside the pores, that will be given by this whole expression. Okay. Or simply in general, it is written like VC minus VO, like total bed volume or total column volume subtracted by the void volume. Okay. So the elution volume or eluted volume varies with the total volume of the packed bed or the total column volume that we looked at previously and the way in which the column has been packed. Okay. Consequently, the elution of a sample is best characterized by a distribution or partition coefficient KD. Hmm. KD is independent of the column dimensions and thus allows comparison and prediction between columns with different sizes if the same resin is used. Okay. So this distribution coefficient of a molecule, which is can be written as or expressed as concentration of the solute in the stationary phase, the matrix or the resin that is being talked about, divided by the concentration of solute in the mobile phase, the buffer that is passing through it. Okay. So the stationary phase corresponds to the pore volume. In case of SEC, the stationary phase corresponds to the pore volume, that is the volume of solution inside the particles Vi, which was the total column volume minus the void volume or V0. Okay. Molecules with access to the entire pore volume will elute at Vt or the total volume, right? So these molecules distribute freely between the mobile phase and stationary phases. Okay, because like it is passing through all the pores of the resin, right? But the other ones, the larger solute particles or larger molecules are aren't really interacting with the pore here, right? Really, they won't be occupying this pore volume. They will just pass through the void volume. Whatever volume is or the space outside the pores are available, that will be used by larger molecules, okay, which have limited access or no access to the pores. So in other words, Kd or the partition coefficient represents the fraction of the resin's stationary phase that is available for diffusion of a given molecule, right? So Ve minus V0 divided by Vi. Okay. So if we simplify it, this becomes Ve minus V0 divided by Vc minus the same expression that we saw previously. And so if Vc minus V matrix will give us the total volume. Okay, the entire pore volume that I was talking about, because matrix is also occupying some amount of space, right, in the whole resin. So if we remove that, then that will give us the entire pore volume. Okay, so this is the expression for this is another way to really write or express the partition coefficient or distribution coefficient. Now see, uh, large molecules passing directly through the resin without entering the particles will have a distribution or partition coefficient equal to 0 because nothing has gone inside. It has just come out through the spaces outside the pore. Okay. So larger molecules will have a there was no partition only. There was no distribution of the molecules. Okay. So this will have a Kd equal to 0. Whereas small molecules having full access to the pores of the resin or the matrix will have Kd equal to 1. So suppose for the salts that we were talking about, it was eluting just before the one column volume of the matrix here, this one. Okay, so for the salts and all, that was eluting out just before the column volume. For this, the Kd will be nearly equal to 1 because it is passing through all the pores of the matrix and then eluting out of the column. Okay. So there will be proper partition and distribution of those smaller molecules in the sample mixture. Okay. Now, SEC resins should be selected so that the important components are found in the most linear part of the selectivity curve with minimum peak broadening. Peak broadening means how what we were talking about, how sharp or how broad the peak is. Depending on that, it also tells about how pure your sample, like separation was, or how pure the eluted or the fractionated sample is. Okay, or dilution. The more the things are broadened, okay, suppose things are in case of the suppose ion exchange, etc., or suppose in case of SEC only, the more broadening is there, that means in one fraction, suppose the see here, what happens here is the wider this thing is, the more broad or the broader this thing is, what will happen is it will elute out slowly. Suppose we are fractionating it at particular volumes. Now, suppose I'm collecting the eluted fractions at around 1 ml. Okay, 1 ml, as like one drop by drop, 1 ml fractions I have collected. So, suppose within 2 or 3 ml, this particular only 3 ml of my collected fractions will have all of this high molecular weight protein, suppose, or molecule. The next one will have, so it is highly concentrated in those fractions, right? But the wider this thing is, so suppose I collected again 1 ml, 1 ml. Now this will go on for 5 ml of fraction, but it will be diluted. The broader the range is, it will be more diluted in one particular fraction. Okay, this is what is being talked about here. So the wider the peak is, there will be, the sample will be more diluted. And so what we have to aim for is minimum peak broadening, minimum dilution, or and minimum time on the column. Okay, the lowest molecular weight substances should be eluted near the total column volume. Okay, so under ideal conditions, no molecules can be eluted with a distribution coefficient greater than 1 or less than 0, right? The steeper the selectivity curve, the higher the resolution that can be achieved, right? If the resin is more selective, then what will happen is it will be able to separate out the components proper. Selective as in it is depending on the pore sizes of the resin or the matrix in question. Okay. Now, if the distribution and partition coefficient is greater than 1, what actually happens is non-specific adsorption happens, or non-specific interaction happens on the chromatographic resin, which actually, so if so the expression is VE minus V0 by VI, right? So if it is greater than 1, that means this thing is lesser and this thing is higher, right? So if that is the case, in that case, there has been, what might be the possibility? One possibility is there might have been some interaction or non-specific adsorption onto the resin, which have blocked the pores, or uh, yeah, which have blocked the pores of the matrix, thereby hindering the passage of the molecules through the resin. Okay, and if Kd is less than 1, if Kd is less than, is less than 0, then there might have been channeling in the chromatographic bed, and the column must be repacked. So what will happen is there is no separation, or like the pores might have been broken, or the pores might have been broadened, so that a particular channel has formed in between, and the things can easily, your sample can easily pass through the column. Then in those cases, you will get a distribution coefficient of less than 0. Okay. Now, in an ideal SEC, what happens is only steric effects contribute to the separation of the biomolecules, and separation is generally insensitive to the eluent composition. Okay, like this should be the ideal case scenario, how what we were talking about previously. Uh, but what happens in non-ideal behavior, suppose when the distribution coefficient or the partition coefficient goes below 0, uh, there might have been some interaction between the resin and the biomolecules. There might have been some adsorption of the biomolecules onto the resin. So this actually affects the separation. Definitely the separation will be affected. And generally only of concern these cases when separating highly charged substances at an ionic strength that is too low, or when separating aromatic molecules on dense resins with a high matrix content, higher dry weight of resin. So what happens is if you are separating like highly charged protein, suppose protein, the amino acids are highly charged, like either positively charged residues are there, or something like that, and the ionic strength of the buffer that you have used is too low, so that the interaction of this, like these charged biomolecules or the molecule of interest, has lower affinity towards the mobile phase, towards the buffer that is having low ionic strength. Okay, so what will be the other way around? it will have preference towards the matrix. It will, because the buffer condition is not really suitable for these highly charged substances to go into. So it will try to interact more with the matrix. So sometimes it might get adsorbed onto the resin. Okay. Or when separating, or if the matrix content is really higher, then aromatic molecules, being like, if the pores are like really dense, okay, the resin has been packed in a higher quantity, or matrix content is really higher, the like while separating aromatic molecules, there can be some non-ideal behavior wherein these aromatic molecules like stay there in the pores and are not able to pass through. Okay. There can be hydrophobic interactions as well in some cases. Okay. So all these can be usually avoided by changing the running buffer. If the buff this all these things will happen only when the buffer condition is low. Suppose there is low ionic strength in the buffer, so that mainly the molecules that is passing through should have, like should be having proper stability, or like should be provided, the buffer should be providing optimum parameters for your sample of interest to stabilize. Okay. So that it elutes out properly, and it doesn't, like, it should be amenable or optimum. Otherwise, if it's not optimum, it will have an interaction with the resin or the matrix, SEC matrix here in this case. Okay. So small biomolecules are more susceptible to interactions because it is having access to all the pores. So it is passing through all the pores, and so it will have more time and more, there will be chances of these small molecules having certain kind of interaction with the matrix, with, or the resins, than larger ones, because larger ones anyways aren't entering the pores, it has limited access. Okay. So the larger molecule will be passing out of the spaces outside, will be passing through the spaces outside the pores. So it usually will be having minimum interaction with the matrix or the SEC resin. And also many times the molecular shape can explain an unexpected elution position. Okay. Now, this was there in the previous class also. So resolution is a function of the selectivity of the resin and the efficiency of that resin to produce sharp, narrow peaks with minimal peak broadening, which is basically, resolution is given by like the volumes, elution volumes of the two adjacent peaks. Okay. Because this will having a higher range, like higher um, elution volume, which means this will be of a lower molecular weight as compared to this one. Okay. And uh, this particular elution volumes, difference between the elution volumes of the two peaks divided by the average of the width of the two peaks. Okay. W1 plus W2 by 2. Okay. This is how we represent or define resolution. Now, homogeneity of the packed bed and of the particles influences uniformity of flow through the column, and hence affects the shape and width of the peaks in the chromatogram. SEC resins having high uniformity and narrow particle distribution result in the elution of molecules in sharp peaks. Okay. SEC resins with smaller particle sizes facilitate the diffusion of sample molecules in and out of the particles by reducing the time to achieve equilibrium between mobile and stationary phase. Okay. Peak width is reduced and resolution is improved. Okay. The more time we give for the smaller particles to really move into all the pores and interact with the matrix, the more broadening will be there, band broadening will be there, and hence the sample that is being eluted out will be dilute. Okay. Sample dilution is inevitable because diffusion is occurring. And to minimize this sample dilution, a maximum sample volume is used within the limits set by the separation distance, that is the resolution required between the peaks of interest. The sample can be regarded as a zone passing down the column. Okay, that is what we were seeing, the bands, okay, of the red, orange, and yellow molecules. So the yellow one was the broadest, right? Like widest one. So we have to take care, if we consider like each, the red zone, orange zone, yellow zone that we were seeing in the previous slide. Then what will happen is, the lesser the broadening is for that particular zone, more concentrated will be the sample that is eluting out of the resin or out of the SEC column. Okay. Now, how do you really prepare the sample and buffers? Removal of particles in the sample or buffers and cleaning solutions is extremely important for SEC. And we looked at few of the things to look at before passing or loading your sample onto the column. You have to clarify the sample before applying it to a column. This will minimize the risk of blockage. There might be particles or some other unknown contaminant that might interact with the resin available. So it will reduce the need for stringent washing procedures and extend the lifetime of the column. Also, suppose some unknown contaminant is there and it has really blocked your column. So what will happen is, usually in case of SEC, what usually people do is you just wash it using water, like filtered, degassed, uh, double distilled water. But if something has blocked the pores or something has happened, some interaction has happened, which is giving unwanted peaks in the illusion profile, okay, or in the whole chromatogram, then what will happen is, you need stringent like washing procedures. Like suppose you have to use some base or high salt concentration, which might degrade the matrix properties, and because the matrix are made in such a way that it should not have any interaction with the biomolecules passing through, but the application of such strong reagents to clean the whole resin or matrix might hamper with the properties or composition of the matrix. Okay, so thereby these stringent washing procedures can hamper the lifetime of the column. So we need to reduce the usage of stringent washing procedures. So for that we have to take care, um, like clarify the sample as much as possible, clarify the sample, the buffers. We have to use filtered buffers, etc., before applying or before passing it through an SSC column. Okay. SSC columns are like really costly columns. So it's always ideal, or it's always expected that you keep it in a proper condition so that you don't hamper the things. Otherwise, your whole experiment, your protein prep, or any other sample prep will be hampered. So what you can do is, we can use high quality water and chemicals. Solutions should be filtered through 0.45 micron or 0.22 micron filters before use. You have to degas the buffers before any SCC separation, because air bubbles, if it is present, can reduce the performance. Bubbles are automatically degassed if filtered under vacuum, and buffers and columns must have the same temperature before use. This is another thing that should be kept in mind, because rapid changes in temperature, for example removing packed columns from a cold room and applying buffer at room temperature, can introduce air bubbles in the packed bed, resulting in poorer separation. And samples must also be clear and free from particulate matter, especially when working with particle sizes of 34 micron or smaller. For small sample volumes, a syringe filter of having a filter made up of cellulose acetate or PVDF membrane can be enough or sufficient. Okay. So this is what is being talked about, the filtration thing here. Filtration removes particulate matter. Wortmann syringe filters, which gives the least amount of non-specific binding of proteins, are composed of cellulose acetate, regenerated cellulose, or PVDF membranes. Okay, so definitely when you are filtering your sample, again that should be that filter membrane that is being used for filtering the sample should, sample or filtering the buffers, that should definitely be something or composed of something which should not interact with the, which should have minimum interaction with biomolecules. Okay, because if things are interacting with the filtration membrane, then that will stick to the filtration membrane, and the things that will come out, like after you filter the solution, suppose filter your sample or filter the cell lysate, or you're filtering the buffer before passing through the column, then the constituent of your buffers are sticking onto the membrane, and that won't really make any sense when you are really passing it through the column of your column or resin. For sample preparation before chromatography, we should be selecting a filter pore size in relation to the bead size of the chromatographic medium, right? So that there is a uh, like the chromatographic resin that is having a particular bead size, the filter pore size should also be having, like, it shouldn't really be, the particles or any other such contaminants shouldn't be of such size that will hamper with the bead packing or the matrix or column like packaging of the resin inside. Okay, because it will again, if something is like the, this applies to all sorts of all type of chromatographic techniques. Okay, if something is interacting with the, or if there is something is interacting with the bead or the pores in case of SEC, so that will hamper ultimately the packaging of the resin and movement of your sample of interest through the column or the matrix. Okay, so we have to be mindful while selecting a pore size for filtration. And so what all are the different factors that can influence resolution? These are the resin related factors like selectivity, the match between pore size and size of target molecule, particle size, particle uniformity. Okay. Uh, if it's more heterogeneous, then the resin uh, should be packed in such a way so that pores of different sizes can be there, so that if the heterogeneous particle or the sample mixture is applied, it can be separated out well, or resolved from one peak to, like, it should be well resolved from one peak to the from the other peak. Okay, because if the particles are not really uniform in size, if there is a lot of heterogeneity between the particles, then uh, it will finally affect the separation and the resolution of the matrix or the whole chromatography. Column related factors that can affect resolution is the bed height and column packing quality. And chromatography system related factors, there are tubing dimensions, diameter and length, because see like we know about the column is there. I think in the lecture videos you saw the whole FPLC system, right? So there are tubes through which the whole sample things are connected from one, like your sample valve is connected to your column valve, etc., right? So the tubing dimensions also matter, and the volumes in the system components also matter. And experimental related factors like the flow rate, how much you have set, and what are the limits of the flow rate. If you go beyond a certain flow rate, it will hamper the column packing. It will put a pressure on the column and damage the column. Okay. Sample volume. What is the viscosity? If the viscosity is more, we will look at it in the next slides. And the buffer composition. Again, it suppose if viscosity is more, the pressure on the column is more, definitely. So the flow rate should be reduced. Suppose in many cases, in many buffers, when this is irrespective of whether it's SEC or ion exchange or affinity chromatography. What happens is, if many times we use, suppose, glycerol. Okay, in the buffers, we require glycerol. The buffer composition has glycerol in it, and glycerol being denser and viscous, it adds to the viscosity of the medium. So what will happen is, as the sample is in the buffer containing glycerol, it is more viscous, and as it passes through the column, so we have to reduce the flow rates in such cases so that it doesn't really put unwanted pressure on the column, which will ultimately damage the column. Okay. See, a critical variable is the viscosity of the sample relative to the running buffer, as shown by the changing and elution profiles of, like this is an example they have shown, hemoglobin and sodium chloride at different sample viscosities. Now, without even looking at the description, we can say that the one that is eluting out later should be the low molecular weight like molecule, and this will be the higher molecular weights molecule. So if we are talking about elution profiles of hemoglobin and sodium chloride, then definitely this one, the red one, will be of sodium chloride because it is much smaller than hemoglobin, and hemoglobin being the larger one will be coming out of the column uh, first, followed by sodium chloride at later column volumes. Okay. So now we were talking about viscosity. If the viscosity of the medium is lower, So this will be well separated. Okay. Sodium chloride is coming out later. Hemoglobin has come out earlier, much earlier. If the things are intermediate, if the viscosity is intermediate, what will happen is there will be, at, or as the viscosity increases, the separation or resolution between the two peaks will be dampened, or like it will decrease. So deteriorating separation caused by increasing sample viscosity. Elution profiles obtained from hemoglobin blue and sodium chloride red were separated. Experimental conditions were identical. Like this is the experimental graphs that have that have been obtained, where they just change the viscosity of the sample by adding increasing amounts of dextran, and due to, like, this is another like molecule that adds in, that makes the buffer, or the makes the medium lot more viscous. Okay. And with high viscosity, we see that the elution profiles are like deteriorating, or there is this resolution and selectivity is decreasing for the resin. Okay. Now let us just see, like, go through this, like we will go through this slide slowly so that we can understand how exactly would one know that, okay, uh, this is void volume, this is the void volume, and this is where my protein of interest. Like we looked at different kinds of peak, right? Like intermediate molecular weight, high molecular weight, low molecular weight. There are multiple peaks. Now, how will you really know that, okay, this is my protein? So all the columns, there are many pre-packed columns. I think in the lecture videos you saw those ones, right? With red endings and white like matrix packed inside glass tubes. Okay, those are like pre-packed columns. You can pack it manually also. So what happens is, based on the dimensions and properties of the type of resin that has been used, uh, samples elute out like at certain volumes. Okay, and that gives us the range of the molecular weights. Like this is the property of the like column or matrix that is being used here. So suppose there are two columns here, which is superdex 200 increase and superdex 6 increase. Okay, this is the naming given based on the property of the matrix, the dimension of the tubing and column. Okay, so these are both resins for small scale preparative purification and analysis with the same high flow agarose based matrix. Okay, where the average particle size is 8.6 micrometers. Now, how do they really differ? They differ in the fractionation range, which results in very different chromatography profiles for the same sample mix. Okay. Now see, this is the profile for Superdex 200 increase, and this is the elution profile for Superose 6 increase. Okay. The same set of mixture, sample mix have been applied, but see, the elution profiles are so different. Okay. So why? Because Superdex 200 increase, this one, gives excellent resolution for biomolecules with molecular weights less than 440 kilodalton. Okay. While larger biomolecules elute together with aggregates in the void volume. So see, as soon as the um, this near the void volume, these higher molecular weights, which are 1, 2, 3. 1 is 970 kilodalton, 2 is 669 kilodalton, and 3 is 440 kilodalton. So this particular volume, this particular resin, which has a certain composition or porosity of the matrix, will really properly separate the biomolecules less than 440 kilodalton. Like suppose this BSA 66 kilodalton, myoglobin 17 kilodalton. This is 1.3 kilodalton or 1.4 kilodalton. Okay, vitamin B12C, based on the sizes, it is eluting out very nicely at well separated peaks. Okay, but it can't really resolve depending on the porosity of the matrix. It can't really, or depending on how, what is the packaging or packing of the matrix, it can't really resolve biomolecules above 440 kilodalton. This is why all these things cannot really enter the pores. These biomolecules which are greater than 440 kilodalton or greater than or equal to 440 kilodalton. So all these can't really enter the pores. It will move through the spaces outside the pores of the resin of this particular uh, matrix, super disk 200 increase, and will elute out in the void volume. Okay, it will form aggregates and will elute out there. But the other one, superose 6 increase, this one, see, uh, superose 6 increase on the other hand has good separation between the largest biomolecules. Okay, see the 1, 2, 3 here, the separation is quite good as compared to this one. Okay, so what will happen here is, now when you are, I was talking about fractionation. Okay, now when you are collecting the eluted fractions, suppose 1 ml of fractions, so this is coming out first. Now 1 ml of that particular fraction will have all these three together. But you are not interested in all these three, you want the third component, suppose. But in the other case, what will happen is the third component will be eluting out at 15 ml volume. Now you have started collecting fractions from here. So 1, 2, 3, 1 ml, 2 ml, 3 ml, 4 ml fractions will have, suppose this one, uh, this first protein, the second protein, the third protein. So the third protein will be present exclusively or in higher concentration, like much higher and purer concentration in the, like around the fractionation volumes of suppose 14 to 16 ml. Okay. So you will have only this component 3 there. Okay, not only, suppose there can be other things, but mostly and in the most purified form and at higher concentration, you will have this third component there. But see, in this case, you will have two as well as one other contamination will be there. So you won't be having this pure and well, like properly concentrated and pure form of third component. Third is ferritin here. It can be some other biomolecule also. Okay, so uh, this is why well resolved peaks are important for you to get pure and well concentrated sample of your interest. Okay, so all these proteins or samples are eluting out at particular volumes. So suppose this is the characteristic of, suppose supertext 200 increase column, that what is the 17 kilodalton protein will elute out, suppose at maybe around 16 to 17 ml. So this is how, if you are getting a profile of your sample of interest and you know that your protein is suppose 18 kilodalton or 16 kilodalton, then it is showing a peak around here, then you know that okay, this is a good range and this is my protein of interest. And suppose you are not getting any other peak, you are just getting, suppose this peak, you know that your protein is 17 kilodalton, so it should be eluting out at around 16 ml or 17 ml of this buffer or column volume, but it is the peak that you are getting is like is coming out at around at like lower column volumes. So that means the mixture doesn't have a protein of interest, or your protein of interest hasn't really eluted out, or there has been some issue that your sample of interest or protein of interest is not there. Okay, so this is how you get to know based on what is the column being used, that whether the eluted protein, like eluted sample or the peak that is being shown is the peak of your corresponding sample or protein of interest or not. Okay. Now see, this is an example of satisfactory separation, where the peaks are well resolved and symmetrical. This is a poorly resolved peak. So what can we really do? Review factors affecting resolution, including the selectivity of the resin, particle size, sample to column volume ratio, the flow rate should also be taken care of, and equipment used. Now, suppose if you increase the flow rate too much, then what will happen is you are not like giving enough time for the samples to really get inside the pores, or in case of, suppose not only in case of SEC, suppose where affinity chromatography is taking place, where the biomolecules have to interact with your matrix. So if you increase the flow rate, what will happen is the biomolecules are not getting enough time to interact with your matrix, or in case of SEC, they are not really getting enough time to get inside the pores and pass through according to their size. Okay, that uh, pressure due to the flow rate, higher flow rate, will might let some smaller molecules to also pass through the void volume and not enter the pores itself. Okay, because the flow rate is too high. Okay, so this way you can flow rate can also affect your resolution. There can be asymmetric peaks where you can see a tailing here. So tailing peaks can be caused by an underpacked column, that is a column packed at a very low pressure or flow rate. Okay. Ensure the resin is evenly packed. There should be even packing. And tailing peaks can also be explained by uneven sample application and interactions with the column. Asymmetric peaks or fronting. Fronting peaks can be caused by an overpacked column, that is a column packed at excessive pressure or flow rate. Peaks appearing before the void volume indicate channeling in the packed bed. What we were looking at, the distribution coefficient less than 0, right? If channeling has happened, then most of the things will come out in the void volume itself. Hmm? And late elution. Sometimes peaks eluting after 1 CV of buffer can be caused by non-specific interactions with the resin. So what we can do is we should always be washing the whole column. There should be a wash step included or extend the elution to ensure elution of late eluting molecules so that you don't lose out on your protein of interest or sample of interest. Changing the buffer composition might also be beneficial. Okay. Now, what can be the different applications of gel filtration chromatography? Um, like you can determine definitely the molecular weight. It is what I was talking about. Suppose for Supertex 200 increase, samples eluting at around 15 to 17 ml or ml of column volume is having a weight of molecular weight of 17 kDa around in that range. So if something is coming in that range, the peak is there, then you can determine your molecular weight from there. Now, the molecular weight and size of a protein is related to the shape of the molecule definitely, and the relationship between the molecular weight and radius of gyration is as follows. Rg is directly proportional to m to the power a, wherein a is a constant and this is like it depends on the shape of the molecule. For a rod-shaped molecule, a is 1. For coils, it is 0.5, and for spherical molecules, it is 0.33. Okay. And you can also see the oligomeric status of the protein. Many times protein tend to oligomerize. Okay. So like uh, when you run it on SDS page, what will happen is SDS page being denaturing, it will linearize the proteins, right? If there are any, suppose if you try to run hemoglobin on SDS page, it has 4 subunits, 2 alpha and 2 beta subunits, right? So when suppose you are running an SDS page, so you will ideally be see two bands, one for alpha subunit, one for beta subunit. And suppose if you are running it on SEC, what you will see, you can see like different peaks, like owing to the oligomeric status of the protein. Okay, if you the monomer will be eluting out at later part, whereas the whole tetrameric part of the protein will be eluting out first. Okay, so like this is just an example. So the native molecular weight determination by gel filtration in conjugation with SDS page can be used to determine the oligomeric status of the protein. So oligomeric status molecular weight, you are seeing something here on the after SSC divided by, because SDS is denaturing, so it will show the things in its monomeric form. So that way you will get to know, okay, suppose something here, suppose in the gel filtration thing, your molecule is eluting out, say suppose here, where the molecular weight is 80 kilodalton, okay, but on SDS page, when you ran the same sample, because and you are seeing the thing as 40 kilodalton, you are seeing your protein band running at 40 kilodalton on SDS page, but on gel filtration, you found that your protein of interest is present here, okay, which is giving us like the range is around 80 kilodalton. So then on SDS page, it is 40 kilodalton, which has denatured, but on the gel filtration thing, here it is only based on size. So that means your protein is forming a dimer. it is in a dimeric state. Okay, if we are talking about protein here, then it is dimerizing there because 240 kilodalton subunits are together, which are passing through the SEC resin. But on the SDS page column, due to the presence of um, reducing agent like PAB and due to the presence of detergent like SDS, we are actually reducing the whole thing and denaturing the whole thing. So the interaction between two subunits are not there, and so you will only see the presence of one band on SDS page of 40 kilodalton protein. Okay. You can study protein folding also. What happens is, so we were talking about denaturing agents like SDS, detergents, right? And urea, guanidine, hydrochloride. So what happens is, as you increase the concentration of urea, your native protein is there, you add urea to it, it will partially unfold. Urea will basically affect the interactions going on inside the native protein between the residues. Okay. As you go on increasing urea, this will be your protein, properly folded protein will be unfolded or completely denatured or something. Okay, here. Now the protein, now if you run this particular whole sample mix through the SEC column, what will happen is the native protein is well packed, right? It will have a certain uh, radius, and then this particular volume being occupied by the well folded protein will definitely be lesser than the unfolded protein, right? So here that is what is being written here. Uh, Protein is incubated with different concentration of urea for a certain amount of time at a certain temperature. Now an SEC column is equilibrated with buffer containing urea, okay, and the incubation mixture is analyzed. So as the concentration of urea or denaturing agent is increasing, protein will unfold with an increasing hydrodynamic surface area, right? Because the compactness is going away, and as a result protein peak shifts towards left, means this side. Definitely as the it becomes larger, the overall size becomes larger, so it won't be, the access to the pores of the resin or SEC resin or matrix will be limited, right? So it won't be able to this suppose this fourth part or the third part will be having problems in getting inside the pores of the SEC matrix as compared to this very well folded native protein, right? It can, because of its limited or like smaller hydrodynamic radii or hydrodynamic volume, what will happen is it can get inside the pores easily as compared to this unfolded one. Okay. So having larger hydrodynamic surface area, at the highest concentration of the denaturant, protein will completely unfold and mostly appear or elute out in the void volume. Okay, because it won't be able to enter the pores, it will come out, but the very well folded native protein will elute at the end. Okay, as compared to the other three stages. Okay. You can also study protein ligand binding. So ligand being small will elute out at the end. And if protein will also have a certain elution profile, but if there is interaction between the ligand and the protein, then you will have another peak here, right? Definitely before the ligand as well as before the protein. All right, because ligand and protein will overall this interaction can will be increase the will be increasing the overall size of the whole sample being used here. Okay. Now affinity chromatography works on the, like we talked about it, on the principle of mutual recognition forces between a ligand and a receptor. Like we have been talking about nickel NTA chromatography, that is a type of affinity chromatography. So what does it really like make use of? The major determinants responsible to provide specificity are the shape complementarity, electrostatic interactions, hydrogen bonding, van der Waals interactions between the groups present on the ligand and the receptor pair. Okay. So this is the receptor R and the ligand. Okay, a mutual interaction between a ligand and receptor forms the ligand receptor complex with the dissociation constant, which is expressed as follows. Okay, individual concentrations of the receptor and ligand divided by the ligand receptor complex concentration of the conjugated ligand receptor complex. Okay. Now this is the same profile like how we looked at ion exchange chromatography HIC. You have to equilibrate your column. Affinity medium is equilibrated in binding buffer. Sample application and wash. You apply your sample under conditions favoring the specific binding, okay, of the tagged protein to the ligand. Unbound material is washed away. Suppose here we are you here the whatever is being shown here is like this particular, this is the, suppose this is the ligand or receptor being attached to the matrix. This is the particular kind of thing which will only interact with a certain type of molecule or certain shape of a molecule present in the whole sample mixture. Now the whole mixture is applied and only the molecule of our interest will be binding to the ligand here on the matrix, or like this the ligand here on the matrix, unless the rest types of molecules will be eluted away or washed out. And then you again put in elution buffer, where either you use another uh, molecule having higher affinity for the ligand here attached to the matrix. So these ones which had formed interactions with the ligand on the matrix, these will be eluted away. Conditions are changed to promote the elution of the tagged protein. In this case, the collected tagged protein is purified and concentrated. The same thing, you first equilibrate the thing, you apply the sample, okay, and then you start washing the sample and then start eluting out, okay. Now, what are the different advantages and disadvantages of tagged proteins and untagged proteins? For tagged proteins, simple purification is possible using affinity chromatography. It is a general two-step purification protocol, and detection of the tag instead of the target protein allows for generic detection method. Okay, solubility and stability can be improved using tags. We were looking at all those things, right? Different kinds of affinity tags in the previous lecture videos. A marker for expression is provided. Suppose a protein is tagged with GFP, then you can easily see the presence of GFP in the eluted volumes. Okay. If the GFP is there in the eluted volumes, the intensity of the color will tell you, can give an idea about the concentration of the protein present in the volume, or the eluted volume. Okay. Or simply the fraction. Some tags allow strong binding to chromatography media in the presence of denaturants, making on-column refolding possible. Suppose this urea thing, something has been unfolded, the protein has been unfolded, then in order to refold on the column itself, there are certain buffers which can really help in refolding of the native protein. What can be the disadvantages of tagged proteins? The tag may interfere with the protein structure. Definitely, it can hamper, because tags are usually having amino acid residues or some other molecule attached, right? So it can uh, add into the interactions taking place between the amino acids of a protein and can lead to some structural variations, or proper folding of the protein might not be there, which will further affect the function of the protein, right? Which will affect the folding and biological activity. Now, if the tag needs to be removed, cleavage may not always be achieved at 100%, and sometimes amino acids may be left. So we were looking at the protease thing, right? Some tagging has been done for purification, but for the downstream applications, you don't really need the tag. So you have to remove the tag. So sometimes this removal of the tag might not be 100% removal. Some of the amino acids might be left. The protease cleavage might be non-specific. So then some of the amino acids will be left, which can eventually hamper protein structure and function. Okay. In case of untagged proteins, definitely the advantage is tag. You don't have to remove anything. Any other treatment need not be given. But purification and detection might not be simple, and there might be problems with the solubility and stability of your protein, and thereby if the solubility is less or the stability of your protein is less, the protein will degrade, and ultimately the concentration or the yield of your protein will be lesser. Okay. See, this is what is being talked about. The tag, if the tag needs to be removed, some amino acids may be left if the protease cleavage is not 100%. The effectiveness of proteases used for cleavage may be decreased by substances like the detergents present in buffers, right, in the protein preparation or by inappropriate conditions. Okay. Now, this is the overview of the sample preparation. We have gone through this earlier also. Suppose some protein is expressing intracellularly, or there is extracellular expression of the protein of interest, suppose. Then for intracellular expression, it can be insoluble in the cytoplasm, it can be soluble in the cytoplasm, or things can be present in the periplasmic space, like in case of bacterial cells. It has cell wall and cytoplasm, right? There is some space in between. So in the periplasmic space also, there are some proteins present. Okay. Suppose you are interested in studying the proteins present in the periplasmic space. So the overall sample preparation will be a little different for each of the cases. Like suppose for proteins which are insoluble in the cytoplasm or soluble in the cytoplasm, the very first thing is you have to lyse the cell, remove the cell debris. Then if something is insoluble, which is not coming into the supernatant, then it goes into the inclusion bodies. You have to harvest the inclusion bodies and solubilize them. And otherwise, if something is very nicely soluble in the cytoplasm, you recover the supernatant and then go for purification. Now, if proteins which are present in this periplasmic space, you have to disrupt the cell wall. You have to remove the cell also because it is not really present in the cytoplasm. And then you recover the clarified sample using proper treatment and then go for purification. Now, the thing is, I am like while telling it's easier said than done because at every step there are a lot of optimizations involved and it takes like it is a little tricky for like till purification itself needs a lot of uh, optimizations and standardizations. But before that, to reach up to the step of purification, there again are a lot of steps wherein you have to optimize at each point to really recover your protein of interest, to check the solubility of the protein, to stability of the protein, where the protein of your interest is going into, whether it is coming into the supernatant, going into the inclusion bodies. You have to make like keep an account of all these factors. And if the thing is extracellularly expressed, something is being excreted out, right? Or secreted out. You basically have to harvest the cell culture medium. Okay. You remove the cell and then recover the clarified sample. So the major parameters while preparing a sample is clarification, stabilization of the target protein. How do you stabilize? You use protease inhibitors. You take care of the pH or the ionic state and add reducing agents or stabilizing additives, etc. Conditions for purification to work. You have to take care of the absorption. Also, you have to keep an account of the available equipment and the sample size, filtration or centrifugation equipment available. And centrifugation and filtration are the standard laboratory techniques for sample clarification. Okay, we talked about all these cellulose acetate and PVDF filters, which shouldn't be binding to, which shows like least binding to any kind of biomolecules, okay, or least interaction with any kind of biomolecules. And then protein stability we have to keep a track of, because in majority of cases if the protein is not stable in the buffer condition being provided, then definitely your yield and output of purification or assays will be really less, and you might not be getting like properly folded or pure protein. You If you get degraded protein, it won't be of any use as such. Okay. And different types of affinity chromatography. It can be bioaffinity chromatography. What happens? Biomolecules are used as receptors present on the matrix, and it exploits the biological affinity phenomenon such as antibody antigen interaction. There can be enzyme substrate interaction or enzyme inhibitor interaction. Okay, for example, GST glutathione. there can be pseudo affinity chromatography wherein a non-biological molecule is used as receptor on the matrix or any sudden ligand on the matrix to exploit the separation and purification of biomolecules. And there are two very specific examples. One is dye-affinity chromatography. What happens is a matrix is coupled to a dye which is reactive, and the matrix bound dye has specificity towards a particular enzyme. Okay. And another case is metal affinity chromatography like nickel NT that we were talking about. So transition metals like nickel, iron, zinc these are coupled to the matrix, and the matrix bound metal uh, can form a multi-dentate complex with protein containing histidine tag, mainly having imidazole ring. The affinity of protein for matrix bound metal is different, and these differences are exploited in metal affinity chromatography to purify the protein. Okay. Now, this is one type of covalent bonding formed, wherein covalent bond is formed, and this is not reversible. For the other cases, it was reversible, right? You were changing the buffer, like the composition of the buffer, and then eluting out your sample of interest, right? Letting it bind to the matrix and then using some changing the buffers so as to elute out your sample of interest. So the binding was reversible. But in this case, what happens is a thiol group is present on the neighboring residues of a protein like SSR. This is the thiol group that we are talking this is the protein, this is the thiol group here, and this is a thiol gel having SSR group, okay, attached to it. And what happens is thiol group, this SH group present on the protein can form disulfide bond after oxidation, right? This H will be removed and SS bond will be formed. See, uh, it removes this particular R group, this functional group, and protein can uh, get conjugated. So bound thiol protein is here. So the protein is basically bound to the matrix here, right? And after oxidation, and when under reducing environment or conditions, disulfide reversible disulfide bond or disulfide cleavage takes place, which is reversible, and free thiol group is given out. Okay. So what happens is the matrix in covalent chromatography has immobilized thiol group, which forms covalent linkage with the free thiol group containing protein present in the mixture. Okay, this one here. After a washing step to remove the non-specifically bound protein, a mobile phase containing compound with a reducing thiol group is passed to elute out the bound protein. And what happens here is then this particular disulfide bond is cleaved because a reducing agent is passing through, and this forms SH, and then this is also cleaved out. So protein is bound to sulfur, and this sulfur here gets reduced to form PSH. Okay. Elute out the bound protein, the thiol group containing compound present in the mobile phase breaks the disulfide bond between the protein and the matrix to release the protein in the mobile phase. Okay. This is how this thing takes place. So a covalent bond has been formed here. Okay. Now for antibody purification, affinity chromatography can be used. You have Cephalose beads. You couple an antigen there, like the ligand attached to the matrix, and then that will be antigen Cephalose beads. Antigen has been attached. Now you have suppose purified your antibody, and so antibody will have specificity or will be binding to a specific type of antigen. You pass it through the matrix, then only the antibody specific to its antigen will be binding, and that's how you prepare your sample. You wash it and then elute out and then regenerate the column. So you get your antibody binding to that specific antigen, and then you purify your antibody. You get the pure form of antibody. Antibodies are proteins basically. So GST based purification, this is an enzyme substrate affinity taking place. So what happens is glutathione S transferase GST utilizes glutathione as a substrate to catalyze conjugation reactions. Okay. So here what happens is cell lysate, you apply cell lysate. There is this enzyme present here on the beads, and protein is yeah, this is like there already present, and you applied the cell lysate having proteins, GST tagged proteins, and then what happens is it will bind to the column, or the beads interact with the beads, and then see, the GST as a tag is purified with glutathione coupled matrix. GST fusion protein is produced, this one, this protein, this and this red triangle is produced by recombining protein of interest with the GST coding sequence, and then the sample is loaded onto the column previously equilibrated with the buffer containing high salt. Now, unbound protein is washed, as you have seen in other chromatographic techniques as well, with the equilibration buffer, and then this fusion protein is eluted with a different concentration of glutathione dissolved in um, the equilibration buffer. Now, purified fusion can be treated with thrombin to remove the GST tag from the protein of interest. The mixture containing free GST tag and the protein can be purified using the affinity column again, as the GST tag will bind to the matrix, but protein will come out in the, or elute out in the unbound form, okay, or in the unbound fraction. So nickel anti-chromatography, we have seen, like you charge the matrix, you prepare your samples, pass the samples through the matrix, and then elute it out using higher imidazole concentration. Okay. Now, what are the steps to regenerate nickel NTA column? You first wash it with, after multiple uses. I think a particular column after like freshly regenerated can be used. It can be used for 3 to 5 times, I guess. And so, how do you regenerate the column? You first wash it with water. You remove the Ni and nickel ions and basically chylate it out using EDTA. Okay, and then you clean it with a high concentration of NaOH, and then again you pass water through the column so that the NaOH is washed out. NaOH, whenever you pass this high concentration of NaOH through the column, it will strip the column of any other contaminant or any other particles bound to the matrix, and then you again apply nickel sulfate onto the column so that nickel is binding properly to the NTA moieties present, and then you again wash with water and store it in ethanol or any kind of buffer, so that, like, for further use you have properly attached nickel ions there, because nickel ions are the ones that will be interacting or having coordinate bonds with the imidazole groups present in your protein of interest, or with the imidazole present in the buffer. Okay. So affinity columns can also be used as a tool to study or isolate interacting partner of a particular protein. Okay, some histidine tagged protein will be binding, uh, to suppose in case of nickel anti-chromatography, if I'm talking about, that will be binding to your interacting with the nickel ions, right? But if that particular protein is interacting with some other ligand or some other protein, then you are not able to really purify that particular protein. Or suppose you want to just study the interaction of that particular protein, then that protein will also be, like, interacting with your histidine tagged protein, and together this can be eluted out. Okay. And finally, you will be analyzing the same thing on SDS-based gel. Definitely for the interaction studies, what you should be doing, you should always be having a control where the other protein is also passed through without the histidine, without including the histidine tagged proteins, so that suppose there is some kind of interaction of the untagged protein with the matrix, then that can also be seen on the SDS-based gel. Okay. Another will be the same thing we read about, like protease cleavage assay. You basically take the thing out, then this particular tag has to be cleaved out, then you give it a protease treatment. You basically cut the peptide out, and then send it for mass spectrometry, and you get the protein of your interest. Like, analyze the protein of your interest. So now let's come to the question answers. Few of the assignment questions. So, a protein was eluted at 15 ml when passed through an SEC column having a void volume of 5 ml and a pore volume of 1 ml, which means VO is or V naught is 5 ml, VI is 1 ml, and VE is 15 ml. Okay, so the KD value of the protein is, can anyone answer? So it will be the eluted volume minus the void volume divided by the pore volume. Okay, so it is 15 minus 5 divided by 1. Answer will be 10. Okay, option B. Now, the radius of gyration is directly proportional to the molecular weight m to the power, this will be to the power a, where a is a constant. So the value of a for a spherical molecule is, can anybody answer? Yes, it will be 0.33. Option B. Okay. Now a reversible binding of a protein with its ligand will not include which of the following interactions? What will be the answer? So, so salt bridge can be reversible, right? Okay. And for covalent bonding, this cannot be broken. For the thiol one that we saw, right? The thiol activated gel and the protein having that SH eluting out as protein bound to SH, like sulfur and hydrogen, then that has formed a covalent bond. Okay. And that is not reversible. So the reversible binding of a protein with its ligand will not include covalent bond. Okay. Option B. The others like van der Waals interaction, hydrogen bonding is reversible. Salt bridge can also be reversible because it's basically between positive and negatively charged residues. Okay. So as you increase or decrease the amount of the like the residues there present there, you can like how ion exchange chromatography takes place. Okay. So basically, you are differing the amount of salt present in there. If the amount is higher, then the interaction of your sample of interest with the matrix is like weakened, and so the higher amount of salt present now will be interacting with the matrix. Whether the matrix is positively charged or negatively charged, depending on that, your and the charge on your protein, final charge on your protein, depending on that, these interactions can be uh, can be reversed. Okay. Now, protein was run under native conditions in SEC and it was calculated to be 120 kilodalton in size. So the molecular weight of the protein in SDS page, everything is denatured here. It was found to be 30 kilodalton. So this is the monomeric form, right? And so then what will be the oligomeric status of the protein? Can you repeat that? Yes, it will be, because 120 kilodalton divided by 30, it will be 4, a tetramer. Option C. Now, a globular protein was incubated separately in 1 molar, 2 molar, 5 molar, and 8 molar urea for 10 hours at 37 degrees Celsius. So, which of the protein fraction will have the least distribution coefficient value? Least means the one which is not even having any interaction with the matrix. That will be the one that will be eluting out first. Okay. So, what will be the answer? Option D. Yes, the 8 molar one will be the one because it will be like most or either completely unfolded or having a higher hydrodynamic surface area. Option D. Okay. Now, in gel filtration chromatography, when elution volume is equal to the void volume, uh, and KD stands for the distribution coefficient. So, what will be the value of KD? if elution volume is equal. Yes, yes. So the thing is, see, the formula is, if we simply go by the formula, it is VE minus V0 divided by VI. Okay. So if elution volume and void volume are equal, that means VE minus V0 will be 0. So your KD will be 0, right? So if we go directly by the formula, it will be 0. Or if we conceptually think about that, what is happening here is elution volume, whatever is going like eluting out, it is eluting out in the void volume. And what elutes out in the void volume are the ones, are the larger particles which are not able to enter the matrix pores. Okay. So if something is eluting out in the void volume, that means there hasn't been any interaction with the porous matrix, right? So there won't be any distribution as such or partition as such on the matrix for the molecules, right? So it will be 0. Now for the smaller molecules, what happens is it has access to all the pores of the matrix. So if it has access to all the pores of the matrix, that means the distribution or partition, it will have a very well distributed or distribution along the matrix, and it will be nearer to 1 because it has access to all the pores of the matrix. Okay. Uh, yes. Okay. Next, which of the following chromatography techniques is best suited for desalting or buffer exchange? What will be the answer? We just looked at different types of gel filtration, right? Like different preparative SEC, analytical SEC, for desalting or buffer exchange. Salts are eluting out at the end, right? So the answer will be gel filtration, option B. Okay. Now the void volume of a gel filtration column can be determined. So we have been talking about void volume, like larger molecules will be passing out through the matrix, okay, as in without getting inside the pores. So how do you really determine the void volume? What happens is you use really like larger molecules or larger substrate so that it won't be entering the matrix and will directly come out without getting inside the pores. So the volume where exactly these larger particles are coming out, then that will give an idea of, okay, this is the void volume of the particular resin in question. Okay, from the chromatogram only we will be able to make out. Okay. So here xylene cyanol is a dye, glycerol is not heavy enough, glycine is also not heavy enough, blue dextran is a larger molecule which helps in determining the void volume of an SEC column. So this is the step to these are the steps to determine the void volume. You prepare the column, definitely you inject blue dextran. Okay, blue dextran you inject and then elute it with buffer at a constant flow rate by detecting absorbance at 620 nanometers, because blue dextran absorbs strongly at this wavelength, and you will have absorbance versus the volume, right? So you will measure the elution volume, which is the volume of the buffer required to elute this blue dextran, this heavy larger molecule, from the column. And this elution volume corresponds to the void volume because blue dextran is too large to enter the pores of the matrix, the SSC matrix or SSC resin. So this is how we will determine the void volume. This is, so basically the elution volume at which this larger blue dextran appears is the void volume of the column. Okay. So these are few of the things. By measuring when blue dextran comes out, we can accurately determine the void volume. Next, we just talked about regenerating the nickel NTA columns, right? Stripping of the nickel from the NTA beads is performed with a buffer containing, yes, it will chelate out the nickel ions. The binding buffer for the nickel NTA column should have a pH of, it will be actually 7 to 8, because it needs to have like it will it needs to have that particular um, charge, the mainly the imidazole group, imidazole ring in question. So 7 to 8 has been found to be like usually this is the range where it will have the appropriate charge to interact with nickel ions. Okay. The GST glutathione is an example of what type of chromatography? Like it is an affinity chromatography, but what interaction has been explored here out of the four? Yes. So which is the enzyme and what is the substrate here? Yes, good. So next, the matrix in covalent chromatography has immobilized. We just looked at the sulfur thing, right? So what will be the answer? Yes, the option D or thiol groups. Okay. Now, the deactivation of the CNBR activated matrix after coupling with the protein can be achieved by incubating the column with. So what happens here is, for antibody purification, this is your polysaccharide matrix. You give CNBR, it reacts with the polysaccharide at a pH 11 to 12, forming this cyanate ester. Okay. And then what happens is, this ester, this cyanogen ester reacts with the amine group on the receptor to form isourea derivative, right? And this amount of cyanate ester is more with the agarose. The protein or peptide ligand with free amino group is added to the activated matrix. So in any kind of moiety, having a free amino group will be added so that this isourea derivative will be separate and the substituted imidocarbonate will be separate. So basically, this is getting deactivated by adding a protein having a free a protein or any kind of biomolecule having a free NH2 group. So out of these four options, which is having a free NH2 group? Yes, glycine is an amino acid. It will definitely have a carboxyl group and NH2 group, right? See, the other ones won't be having NH2 group, like free NH2 groups. Okay. So option will be glycine. Option A. Okay. In the carbonimide mediated receptor coupling, the receptors are coupled with the matrix by a dash linkage. So, see, this is the polysaccharide matrix. This is carbonimide, and then by reaction with isourea ester, this dialkyl urea is separated out, and your receptor is bound to the matrix here. So carboimides reacts with the matrix containing carboxyl group to form isourea ester here, and the activated matrix is then allowed to react with the receptor molecule having a free NH2 group. Okay. Receptors are coupled to the matrix by a secondary amine linkage. Okay. So the answer is very direct here from the picture. Answer will be option C. Okay. And the last question is, a protein A has a DNA binding site. So, which of the following receptors should be present on a matrix, or what should be coupled to the matrix to be used for capturing this particular DNA binding protein for affinity purification? Now, these are some of the ligands that can be attached to the matrix which has affinity towards these kinds of proteins. Okay. So suppose lysine can be attached to the matrix which can bind to ribosomal RNA. Lectin can be attached which can bind to glycoproteins. Heparin can be attached which can bind to DNA binding proteins. So the answer will be option B here. Okay. Like this is the direct table from the lecture video only. Okay. So that's it for today's tutorial session. We will meet again the next Sunday for the lecture 9, like week 9 lecture videos. Okay. Thank you all for joining. See you in the next class. Okay. Thank you.